interface, but also try to understand that this is just an interface where we need to download a PDB file. So the PDB file is the structure where it's deposited with all the coordinates, and the PDB is usually found in the RCSB website. So let me show you the RCSB website. So this is the RCSB website, as you know, and we can basically select any PDB here. So I can, let's say I'm going to take ubiquitin, or if I remember the PDB that it's called 1UBQ, I can just look at the PDB here. So when you see the RCSB, you see all the information of the PDB, but you can also download the file. So you can download the PDB right from here and have that in your folder, and then you can open it in PyMOL, okay? So open a file will be file, and then you can, in PyMOL, you can do open, and then open the file in the PDB. Alternatively, what I want to show you is how to use the command line to fetch a PDB file. So the way we do it, we basically go here and write fetch. And then after writing fetch, we can fetch the 1UBQ. So we can do 1UBQ and just press enter. And if you do that, do no capital letters and just press fetch space 1UBQ. So just to know, I don't see your faces, but everyone got it?
Yep, got it.
Okay, great. So, so what we can see here, you can see the structure of ubiquitin, and this structure was reported, remind you here, that was actually pretty old. It was 1987 in a 1.8 angstrom resolution, and the method was X-ray diffraction. Here you can see other related description of the statistics when we use the PyMOL. So we're going to use the 3 buttons of the mouse. If you don't have a 3-button mouse, there are some features that we cannot do. Okay, so for once, if you just press on the left click of the mouse and just keep it press, you can move the structure from one side to another, and you can actually do whatever you want with it as long as you keep it pressed on the left click of the mouse. Everyone can do that?
Yeah.
Okay, good. The right click of the mouse, if you can take it and keep it pressed and going up and down to the sides, or up and down, or to the sides, you can zoom in and zoom out what you see, okay? So that the left click kept press, you can move it. The right click kept press, you can zoom it. Everyone can do it? Okay, good. The next thing that we can do is we can use the middle button by pressing the middle button on a certain part. Let's say, look at these red dots. These are water molecules. Press with the red dots on one of them, you can orient towards this red dot. Everyone have a mouse that they can click on the middle scroller button and just press. You can center on this dot, or it's amino acid or whatever. But for now, it's just the dot. So everyone can do it. Can zoom in on one and just move it around. So once you move it, the whole that's going to be the center. Everything going to be around this part. Okay. Now, if I want to go back to the original, always if I want to go back, I can always press here, reset or orient around this place. So if I press reset, it will just bring me to the original side. Or if I press orient, it will bring me also to the center, okay? So these are also possibilities, okay? Everything that I press with the middle mouse, you can see it actually highlight here. It will tell me, you click now, you want UBQ on this chain and this HOH, which is H2O, and number 97, okay? So now we can see it here. So for now, anyone has any questions about what happening here? Anyone had problem to do these maneuvers?
No.
Okay, good. So now what I want to do, I want to try to dive in a little bit, a tiny little bit farther. So we fetch a protein, but now I want to clean out one thing. I want to remove the water molecules now. just to make it clear. So now I want to show you this panel here. And this panel, you see A, S, H, L, and C, okay? The A mean action. So when I press that, there are different actions that I can do. The S is show, is what do you want to show, okay? The H mean hide, what do you want to hide? And L mean label. Do you want to label a certain element or amino acid? And color is just color because afterwards, this is all graphical representation. So we can color different things. So in every session, in every PDB in every PyMOL that you have session, you're going to have this all, which means that you can do this to everything that you're going to have here. Let's say if I'm downloading more molecules here, or you can just work on your protein. So if I press this one like that, it will disappear. And if I press it again, it will appear, okay? Try to make it appear and disappear. Now, if I want to hide this water molecule, there are two options that I can do that. There is one option that I can do it by using the hide waters. Or I can go to command and I can say remove residues. Now, when I say residues in plural, it's RESNHOH. So yeah, maybe I'm gonna write it in the document on the side, so you guys are gonna have this. But essentially, every time you want to remove something, you can just write remove, and RESN is what residues in plural you want to remove, okay? And HOH. Alternatively, I can just go to hide waters. So many of the things that I'm going to show you with the command line is cool because you can do more defined selection, but many things you can actually use this part to change some stuff. So now I basically remove all water. So now I have the protein without the water molecule that was solved with it, okay? So this is the ubiquitin, okay? So the next thing is, let's try to understand what's happening with this structure and how we can change different elements. So one thing I want you to pay attention, so here there's this panel that's talking about the different parts of the things that I can change. And here there's also what is the mouse mode that we are using. The mouse mode that we're using is 3-button and we are on viewing mode, okay? Keep that on viewing mode unless we're going to do another session that is more advanced that we can change this to editing. So if I just click that, it will be edit. This is just viewing, okay? If by accident you press on that, you can change some of these things that you can do with the mouse, okay? But essentially you can see everything that we have happening. This is left click, middle click, right click, and wheel, okay? You can see what happened if you press left click plus shift, left click with control, left click, control shift, and so on, okay? So you can see that if I just press the lift, I rotate, okay? If I press the right, I move zoom, okay? That's what it's saying here. Now, what happened if I press the middle, okay? In the middle, I can move the structure from one side to another if I just keep it pressed, okay? Try to do that. Try to play and be very comfortable with your left click, your right click, and this one, okay? So that's one way to do that. The other parts are important. In this mode, we're selecting only residues, but we can also select chain, we select segments, objects, molecules, so on, okay? So keep it as select residues. It makes things a little bit more easy. At the button panel here, there are different modes that you can see, like start, play, like a record thing. That's actually important to make movies. But what I want you to press is I want you to press on S. So the S is known as sequence. Press on S. Now you can see the sequence of ubiquitin. Ubiquitin is 76 amino acid. You can see the sequence of ubiquitin. So this is very important. Where we want is sometimes oriented. Where is the C terminal? Where is the glycine of ubiquitin? If I press on the glycine, it will see that the glycine is here. It's right here. This is the C terminal. Try to press now on the N terminal, on the methionine. Press on the methionine. Where do you see it? The methionine is here, right? Now you see that we just see like dots. We don't see actually the chain. And the main reason is that by default, the PyMOL only open in cartoon mode. So basically we only see cartoon. So let's just see now what happened if we change to show the amino acids. So let's take a look at the amino acids. So show amino acid will not be cartoon, there will be sticks. So we can press sticks, and now we can see all the amino acid of ubiquitin. So again, go to the panel of ubiquitin here, to the line of ubiquitin, press on show, and press on sticks. Now you can see all the sticks, and this is the amino acid of ubiquitin. Okay? Now let's hide the cartoon and just look at the amino acid. So we're going here to, instead of show, we just go to hide and hide the cartoon. Hide the cartoon. Anyone have any problem to see what I see now? Everyone see what I see now?
Yeah.
Why?
I didn't see it.
Okay, so what do you see now, Why?
I'm seeing another pictures. Didn't like yours.
You just see the amino acid? You see the amino acids? Like
No.
that?
No.
Okay, anyone else that doesn't see the amino acids?
What?
Everyone see that? Only Why? Okay, so Why, I want you to do the following, okay? And just press on hide everything.
Hide everything. Yeah.
And now show sticks. So the sticks are somewhere here.
Oh, yes.
Now you see what I see?
Oh. Yeah.
Okay, good. Okay, so now we see the amino acid of ubiquitin. Okay? Now that's kind of cool. Okay? And we can learn a lot of that if we're interested in certain parts, right? But what I want you to do now, I want you to select only few amino acid, and we're going to create a selection, okay? So there are few ways to do it. I want to show you the GUI way, the friendly way to do it, and I want to show you the way to do it in command, okay? So this part always will be, once we press something, that's going to be press a select, okay? If you press the select, you probably find the first amino acid that we select before, but this is going to change every time. Two things that you need to remember with PyMOL. The undo is not working, okay? So everything that you do, every progress that you do, you better save it as a session. We're going to get to it. But now what I want you to do, I want you just to select the last two amino acid of ubiquitin, okay? So the way to do it, you can either go to the structure and say, okay, I'm going to select this amino acid and this amino acid. Sorry, this amino acid. So just by left click on this amino acid, it will select it. And you can see here on the sequence that it is selected right here. Now, if you press something else, if you press it again, it will disappear. Okay? So that's one way to do it. The other way to do it is go to the sequence and just select these two amino acids by click on them. So it will still select these two glycine, GG or glycine, okay? The other option is we can go to the command line and say select residues, in this case, 75, 76. Oh, here. So that needs to be clicked. So that's the other option. So it's gave it the select basically means select. It's just abbreviation of that, and it's gave me that. So these are the three options that you can do to select residues. Okay. So now I want to color these residues, let's say in red. Okay. So I don't want to color the whole structure in red. I just want to color these residues in red. So I can go to color on the select here and go to the color and change that to red. Okay? Everyone were able to change the last amino acid into red? Okay. Now let's go to action and rename this selection. So you go to rename, and here there's another place open here, and it's saying select. We can delete the select and just write, let's say, last residues. Okay? Or write GG or whatever. So now this is found here, and now we have that in the selection. Okay? So now we selected a certain amino acid. Okay? So what I want you guys to do now, I want you to go back to a cartoon mode. Okay? So I'm going to give you 2 minutes or 1 minute to change everything to cartoon mode, and I'm only talking about the ubiquitin, the protein itself. I don't want to see side chains, I don't want to see sticks, just cartoon, okay? So let me know if you got it. Who got it?
I've got it.
Okay. Sam got it? Where's Angelica? Oh. I don't see everyone. Okay. Everyone got it only in cartoon? Natalie?
No, but don't worry about me. It's hard for me to follow along because I have to keep switching between screens because I only have one laptop.
Oh, okay, okay. So I'm going to do it. So now I want to hide all the side chains. So I'm going to hide all the sticks, and I'm going to just show the cartoon. Okay? So I don't want to see the side chain, I just want to see the cartoon. Okay? And you see that our selection is still colored in red. Okay? So this is the last C-terminal of ubiquitin, which is super important because this is where it's going to attack the other ubiquitin, and you have a ubiquitin chain. Okay? So now what I want to do, I want to explore how many lysines we have in ubiquitin, what are the lysines, and how can we select all the lysines in one simple command, okay? So to do that, first of all, I want to change the background to a paper style background. So we usually publish papers in a white background. So I want to change the background. There is a command line to change the background, but in this way, we can just go to display. In the display, you can go to background, and in the background, you can change it to white. I'm just going to stay here to you guys to find it. So go to display and change the background to white. Everyone got it in white? Good. Okay. So now we see the structure of ubiquitin, okay, on a white background. Now, I want to select all the lysines of ubiquitin, and the reason is that the lysines of ubiquitin are critical because they will tell us basically where can have a ubiquitin chain form, okay? So to do that, I need to actually open a new selection, right? So there are a few ways to do it. So one way to do it is do it manually. If you have such a small protein, you can just say, okay, I'm going to select this lysine, and so on, okay? But what if you have a complex of thousands of amino acids? How you do that? So to do that, we just press on cell, lysines, comma, RESN, LYS, lys. I'm going to repeat that again. You press cell, SEL, the SEL will form a new selection line here. We call this selection line lysines. You can call it whatever you want. And then this comma is important because then it tells you what do you want to select, what do you want to select within this selection. And then you write residue RESN, meaning that it's more than one residue, and space and LYS. LYS is the 3 letters for lysines. Okay?
Do you have to use the 3 levels, or can you use the single?
You have to use 3 letters residue to do the residues, yes, in PyMOL. So, for example, I want you guys to do exactly the same thing, but now I want you to select all glutamic acid. Try to select all glutamic acid. Just to give you an glutamic is E, so you just can say glutamic, okay? Try to select only glutamic acid.
Is there a reason why you previously wrote select and now you wrote SEL?
Yes, SEL, you basically open a new part here, right? So it's going to be, I'm going to give a new name for the selection. Everyone got glutamic acid highlight? Yes?
Yes.
Okay, great. So now I want you to, so let me do that. So cell, I'm going to do, let's say, just call it glut, comma, RESN, glue. All right, so now I found this. Now I have this part and I have this part, okay? Now, what if I just want to do, let's say, arginine and aspargine, okay? So how do I do that, okay? So I'm doing exactly the same thing. Let's just call it acid. So I'm going to do SEL, let's say acids, and just going to do, what did I say? Let's do aspartic, ASP. So I do ASP. Oh, sorry, select acid, and then RESN, okay? So exactly the same, but instead of choosing one amino acid, selected only the glutamic and aspartic, okay? So this is a powerful way when you have something that is very, very big complex and you just want to focus on certain amino acid, or you even want to find your amino acid, right? So that's one way to do it, instead of looking at a whole long sequence. So you can just use the command line to do it, okay? So now what I want to do, I want to start at the moment, just remove everything that there. So basically unselect everything. I just want to focus on the lysine. So press on the lysine and just show the side chain. So how do we show side chain? We go to show, and instead of choosing sticks, which will give us the whole backbone of the amino acid, we can just show the side chain in sticks, okay? Again, I'm going to show on the place of the lysine, going to side chain, going to sticks. Now click on somewhere else, and I want you guys to appreciate what's happening here. What's happening here? These lysines are the most important lysines in ubiquitin biology, right? But one lysine is mostly important, is the lysine 48, okay? Try to identify where is lysine 48. So how do we identify a specific lysine? Without selection, we can either click on each one of them and see what highlight here. We can go to the sequence and see, okay, this is lysine 48. So this is lysine 48 right here, okay? This is one option, okay? The alternative option is to click on each one of them, but another option is try to label them, okay? So how can we label each of these residues? We're going to the place where we have the select here, and now we're going to label. So press on label, and when we press on label, we see different options. So we can have residue, and we can have residues one letter, and so on, okay? Residue name. And let's try to do residue one letter, okay? I don't know if we don't see that because it's white. Let me see. Let me try this one. I don't know why I didn't see that before. Okay, so now we see the K48, K63, and so on. We can do that for all of this, right? So we now can see the amino acid, and we can see exactly each of these residues that can be attacked by another ubiquitin. So essentially, a K48 will be another ubiquitin that will come with the C-terminal here, and will make a polyubiquitin chain right here, okay? So I want to do a pause now before I'm going to move on to the next thing, and just let you know, this is very, very, very basic, right? So there are so many things you can do, but the reason I'm giving you this idea is just now it's really up to you how you want to take it. You can do so many things here. You can change how this protein will look like, whether you would like to do it in cartoon, or I can even look at only the backbone. So I can only show the ribbon. The ribbon is only the backbone. So if I hide the cartoon, I can just see only the ribbon. So this is what we call the C-alpha backbone of the protein. Okay. Sometimes it's very useful to look at that like that. When we look at receptors, it's sometimes easier to look like that for the eye to analyze that. So I want you to, when we finish this PyMOL session, or whenever you have the chance to explore a structure that you are interested, try to play with these different ways to look at the structure, okay? One very interesting way that we like to look at structure sometimes is look at surface presentation. So I'm going to now hide everything, and I'm going to show only the surface of the protein. So you go to show, and you go to surface. Okay? Everyone got this blob?
Yeah.
Cool. So this is more or less how protein may look like, right? You can color it in a really cool color, and you can make a figure out of it, okay? So before I move on to the next thing, I want to just show you that. Let's color it in a really cool color that you like to color the ubiquitin. And now let's make a figure out of it, okay? How do we make a figure now? There are two options. One, you can go to File, and you can go to Export Image as whatever you like, usually like PNG. The other thing that usually I like to do for figure for paper is to make sure that I'm in high resolution and not the default of PyMOL. To do that, I need to press this command, okay? I write ray, R-A-Y, and try to understand what is the frame of the figure that I want to have. I mean, you can imagine that the figure can be, you know, this small if you have other things going on, or this big, and so on. But let's say you center it. Let's say this is what you want. So I can say ray 800 over 600. So I get something like that. There's a little bit misspaced, but what you can see when you press ray, you can see that there's additional graphical shadow that are added here, and that's kind of cool. If you don't touch anything and just keep that like that, you go to File, and then you export this image as a PNG. I want you to export this image as PNG, and when you save this image, use that with opaque, with transparent background, with transparent background. When you use transparent background, you can put it in your PowerPoint on any background that you want to do. So again, I did the ray to make this structure sharp. I'm going to File, export image as PNG, and then here, ray trace with transparent background. And okay, good. So let's go back to the structure. Just click somewhere. By the way, I want you to know that if you press escape anytime, you just see the command line over here, and there's some people that can go only to see the command line. So you can just see all your history file. So press escape, you just see this one, this is your command line, and this is the frame. Anytime that we do a more substantial work, I usually recommend to go to File and save the session as, right? When you save the session, you can always go back to the exact session and where you left it, okay? Otherwise, it's very hard to undo. So every now and then, I like to save the session. Okay, now what I want you to do is bring this one to a cartoon and color the cartoon in blue. So I don't want to see the surface, I just want to see the cartoon and color the cartoon in blue. Let me know if you got it.
Yes.
Okay. Okay.
Yeah, I have it.
All right, perfect. So I'm going to do it. So I'm going to hide, I can hide everything, and I guess I can just show a cartoon, and I can color it now in blue, and I'm going to do it in light blue. Okay, so now we have this really cool image. Okay, now we have also let's select again the license. Let's take a look at the license. You can see that we don't see the amino acid again. We need to show sticks because I hide everything, so I can show the side chain only. Let's take a look at the side chain. And now I want to color in blue all the lysines, okay? So there are two options we can do. We can just go here and color blue, but I want to show you the command line because it's easy. So just color the lysines. We can say color blue, comma, lysines. Okay? Same things. It does have this effect on this one, which is strange, but yeah. Everyone got that?
Yeah.
Okay. So what if we have a structure, and within the structure, we are interested to see a certain amino acid, one amino acid out of like 200 amino acid structure. So we can select this one amino acid, right? Instead of just saying lysine or something like that, we can select a certain amino acid. So choose whatever amino acid you see here, whatever number that you see. Let's choose, I'm going to choose I36 right here, okay? I'm going to choose this one. And I'm just going to select that. So I'm going to press cell. So that's going to create this selection, I36. But I can call it whatever. I can call it whatever I want. I just want to say I36 because this is how I select that. And I'm going to press comma, space. And I'm just going to pick up one amino acid. So it's not going to be RESN. It's going to be RESI. Okay? Space. And because it's only in this chain, it's going to be 36. And I just selected I36 right here. Okay? So this is how you select a certain amino acid. Okay? So the last session I want to do with you, I want to do superposition between two structures. How can we compare now between this ubiquitin and another ubiquitin that was published somewhere else? Okay? Or for example, if you're working on ubiquitin ligase 1 and you want to compare between ubiquitin ligase 1 and ubiquitin ligase 2. It doesn't matter. We're going to use that a lot. How do we do that? So we first need to fetch a new PDB, okay? So let's fetch another PDB of ubiquitin. And let me see if I have something. Okay, so we're going to fetch the following. We're going to fetch a K48 DiUbiquitin chain, okay? So the PDB is 1AAR. So let's fetch it. It's fetch space 1AAR. Okay, now we have two structures. We have our structure that is right here, and we have these two structures that are right here, okay? So what I want you guys to do now, I want you to unselect the one ubiqui, so we're only going to see this diubiquitin, okay? Now, let's hide the water molecules. You can use either the command, as we talked before, you can just go to hide water. Everyone have this this structure in front of them?
Yeah.
All right, cool. Now, let's try to do one more investigation only on this structure. So this structure recapitulates a really, really important polyubiquitin. It's not polyubiquitin, it's actually diubiquitin chain, but this is the first two ubiquitin chains that happen. And if you remember, what happened here is that ubiquitin covalently bound to the other ubiquitin by forming an isopeptide bond with an epsilon group of lysine, okay? So I said lots of stuff now, right? I said an isopeptide bond with an epsilon group of lysine, okay? So if any of you need to refresh, what is it, how amino acid look like? What does it mean, an epsilon group? Epsilon group is a side chain, right? We have, one is one of the side chain. So in the ubiquitin field, the glycine 76 of one ubiquitin, of the first ubiquitin, will attack the second ubiquitin on its lysine, okay, and form its isopeptide bond. So how is that going to happen here, and what do we see here? So first, I want us to identify where is the lysine 48 that get ubiquitinated. So let's select and try to identify the lysine 48. Try to do it if you remember how we did it before. If you don't remember, you can press escape and try to see what we've done before here. Try to select lysine 48, and you can do it with whatever method that you want to do. And let me know when you're ready. So only in this structure, select the lysine 48. And thumbs up when you're done.
Done.
Anyone else?
I think I'm done.
Okay, good. Leo, Angelica, you with me? Okay.
Yes.
Okay, so let's take a look at these two lysine 48. So I'm going to use these in the second chain, okay? So let's keep this one. And now I want you to pick up the glycine 76 of the first ubiquitin, this one. So because we didn't do anything, this is all going to be under select here, S-E-L-E. So everything that you do that you are not renaming by default will be S-E-L-E. I want you to show that as sidechain stick and color it in red. Being a sidechain, do the show all sticks, like show, go to select and go the select that you do and do sticks here, just to stick. So you will see the covalent bond. Now, I want you to use the middle mouse button and just click on this one of this amino acid and zoom in and see if you can see what I see. You can move with the middle one. If you keep pressing it, you can move it from one side to another. But I just want you to zoom in on this bond. This is the isopeptide bond of ubiquitin chain. Everyone see this, what I see? Let me know if we need to go over it again. Everyone see what I see? Okay, I'm going over it again. So basically I select 48 and glycine, that was here in select, and I color that in red. I show that as sticks right here. And now I see this link. This is a linkage. It's also known as K48 linkage. Anyone has problem to see that? This red color just basically put everything in red, right? But I can also go to color and go by element and change it to this type of color. Okay, so this color is by element. This is more convention way to color amino acid. Now the isopeptide bond is between glycine 76 and the epsilon group of lysine. This is the epsilon group of lysine. This is basically the final end of the side chain. This is super unique in biochemistry. It's not happening. Most of peptide bond happen between carbon alpha. There is almost nothing like that that happen at the end of the side chain in the epsilon group right here. So it's very, very unique covalent modification. And this modification is strong. You cannot break it no matter what. You can boil it, you can put it with SDS, you can do whatever you want. This bond is solid like rock. Okay? So, and you can imagine that this is just the first two ubiquitin, but then again, the ubiquitin chain will build up, and this is going to be where it's going to bound the substrate, right? So you have the first here going to be the target, and then you have the polyubiquitin chain. And this polyubiquitin chain will have a really interesting surface that can be recognized by the proteasome, okay? Let's do now, the last thing that I want to do is I want to do surface presentation on that, and then I want to superposition the first ubiquitin with this. So let's first do just a surface presentation of the diubiquitin chain, okay? So go ahead and do it. Surface presentation without anything else, just this. Just the surface presentation of this diubiquitin. So the way we do it, we go to we can do hide later. We can just do show surface. We can hide everything, or we don't have to because nothing is bound, but we can do that. And let's color that in whatever color that you want. It's going to look like a big bulk of something. But now you can imagine that when you have more ubiquitin, this is going to be a really long bulk of something that can be recognized by the proteasome. It's very defined structure. Everyone got this? Yes?
Yeah.
Okay. Last for this session today, I want you to hide everything. And I want you to show cartoon only, and you can use all. So all is basically, you can show all cartoon. So we basically do cartoon and highlight the one UBQ. So you will see it like that. Let me know, please, if you have similar image.
Yes.
Kara? Yes?
Yep.
Okay. Okay, Sam, Leo, you with us?
I have something vaguely similar.
Okay, that's okay. It can be sometimes it's random, right? Doesn't matter how it looks. But what you can see here, that you can see two structures basically are putting together. They are totally independent, but the graphical mode cannot really separate them, as you see, right? But so once I have a structure that are fetched, as you can see here, I go to, I'm going to command line, and I'm pressing the following. I'm typing the following: align, A-L-I-G-N, space, align. So now I want to superimpose. And the most important thing is, what is the moving molecule, and what is the molecule that is the reference, okay? I want the DiUbiquitin to be my reference point, and I'm going to superimpose the one ubiq, the first ubiquitin, on the two ubiquitin, okay? So I'm going to press align, one ubiq, that's the one that's going to move, comma, and I can either press 1 AAR, or I can just do the initial and press tab. It will complete it. So tab is actually really good to complete some stuff, okay? And then I press enter. Oh. Okay. Who got the alignment right?
Me.
No. Now we can analyze different features. We can say, hey, one ubiquitin, when it's bound in a polyubiquitin chain, there are some stuff that are not perfectly aligned, right? You can say, well, this loop might be a little bit different, and maybe this loop might be different. But to be honest, there's not much change with ubiquitin, because in crystallography, there are some very minor changes between different parts, and that's okay. You can see that this angle here probably happened because now it's creating the bond, and here there's no bond, so it's more free. And we know that this C-terminal is pretty flexible, okay? So that's it for today. One thing that I want to tell you that I forgot to say is that you can always go, when you look at selection, if you go to PyMOL here, you can press help, space, select, and you can get all the commands that are important to do different selection, okay? So in many things in PyMOL and Python, you can do this help button to get most of the commands that relate to what we talked about, okay? So that's one option. I'm also going to put a very small paragraph of what we talked about, and I highly encourage you guys to look at structure that you have and try to play with it using PyMOL. Nothing will happen. You can do whatever you want. You can save it. You can move it around. This is really the tip of the iceberg. There's so much we can do. There are ways here that we can measure. So here there's like a measure. You can measure between here, and you can see exactly what is the distance between these two proteins. We can generate asymmetrical, symmetrical copy mates. Is basically understanding how this structure was, how the crystal was formed by going to generate a symmetrical mate. Okay, so I'm doing something like that. Will give me basically the way that these proteins were arranged in crystallography to make the structure. So you can understand that we only look at one copy that represent all the copies, but it's all symmetry. So this is all things that we can do. And sometimes it's important for some of the structure to understand how they crystallize, and whether the packing between the crystals, between the proteins may interrupt or may change some of the loops that might seem as different. So we always want to try to see this part. So that's just different ways that we can look at structure. But the cool thing is that you can really make really cool images from that. And you can always go to do the what I like to do, that it's the saying rock. You can go to press rock right here, and now you can see that your structure is moving. And it's kind of cool, and you can always save that as a video. So you can go here and save and export movie as MPEG. So if you have a cool structure, you can show that how it moves from one side to another. And this can be actually a better design in the different ways that we can move that in different ways. So I'm not going to do like how we can present that in a movie, but just know that you have different feature to show that. So that was just an entry-level idea to PyMOL, how to fetch a model, how to use the mouse, and how to color different parts, how to take advantage of this software, and just give you a really flavor of what can you do with this one. So we did a little bit, play with how to look at different features like cartoon and amino acid. We're able to select different parts and color them and save an image. So all these are very, very powerful, and I highly encourage everyone to really get as much experience with this. There's so much you can do. There's so many things that I don't know in PyMOL that can be done, and every time I'm surprised to see that there are more things that people can make out of it. So it's kind of cool. I'm gonna again put this document. In this document, there are two very important links. One is the practical PyMOL for beginner, and the other one is PyMOL wiki. Both of them are great. There's also a great tutorial. So that's most of the things that that I want to talk about with this thing. So any question about this session, or any ideas, anything that you want to ask?